So it's my pleasure to welcome our opening speaker, George Church, Harvard and MIT professor, founder of many companies, and Nobel caliber scientist. Welcome, George, to our conference for the third time. It's an honor.
Thank you very much. I will touch upon many of the topics today, including neuro and devices, but it will be in the context of aging reversal. And we say aging reversal rather than longevity for reasons will become evident in a moment. My conflict of interests are on the web. They don't fit on the slide anymore. I'm very thankful to those organizations as well as these people in the Personal Genome Project, which is worldwide, meets about a couple times a year. These are some of the countries involved, and some of the agencies that have used our products are NIST, FDA, and NIH ENCODE project. We provide, as far as we know, the world's only open access data and cells. And the data is whole body level all the way down to cutting edge molecular imaging, which you'll see in a moment. Almost every experiment I'll describe that involves human beings or cells is through this project. And here's just PGP is that Personal Genome Project again and again throughout high precision whole genome sequencing, CRISPR, and so forth. These were all developed in that context. And the technologies we developed have gone faster than most, faster than Moore's Law by quite a bit, especially since 2003, where multiplexing really kicked in, molecular multiplexing. And we've improved both reading and writing DNA about 10 million fold to the point where there now are viable business models for $0 genomes. For example, Nebula and DigiD8, where the tiny costs, the cost down from $3 billion to $300, that tiny cost can be defrayed by either the research market or the payers for serious Mendelian diseases, which I estimate is $1 trillion a year. This multiplexing, the digital gearheads will get this immediately. Molecular multiplexing is like communications, where you can have 100 to 1 trillion reactions in a droplet. This is not parallelism or automation. It's actually mixing samples in such a way that have been barcoded, and you can tell them apart. And so the cost of doing a reaction, of doing 1 trillion reactions, is about the same as doing one reaction. Here's a cute device category, not just nanopores, but you'll see molecular electronics in just a moment. These are single molecule methods that we've been working on for decades, since the 80s. Here's one where we fused a polymerase to a pore. This doesn't happen in nature very often, but we engineered this, and you can get baseline separation of single nucleotides, triphosphates, and you can even distinguish runs of G's, A's, and T's because of this baseline separation. Now that's a nanopore category, and we worked with both Genia and Oxford Nanopore. A new category is molecular electronics. We've been talking about this for decades, but now we have one in the form of Roswell. It's still basically in stealth mode, but it's the source and drain and gate of a transistor. The gate is now polymerase. And there are a few patents on this. This should be able to hold 100 million DNA sequencing devices. DNA sequencing devices used to be the size of large refrigerators. Now 100 million of them will fit in a square centimeter. So back to aging reversal. It has been a lot of wishful thinking and not terribly good science ever since ideas about fountain of youth, wishful that you can just something you eat or don't eat. And so I'm involved in some efforts to do clinical trials on things that that are nutritional supplements or used in some other field, including probiotics and nutritional supplements, which I think all of these things should be going through clinical trials, and they are, if I'm involved. But a more serious set of wearables, if you will, wear inside your body have to do with cell therapies and gene therapies. And here are some, one of the ways that we're making human organoids so that they're getting closer and closer to organs, and I'll show you actual organs in just a moment. But these organoids can be very rapidly prototyped in the order of days at near 98% efficiency, and they are now vascularized. So this is work of Mark Schuyler Scott and Jennifer Lewis, who we've collaborated with on making endothelial cells, not just getting them to mix, which they didn't used to do, but they make tubules, capillaries, that are very similar to the native ones. These are 5-micron inner diameter, and when we put them into animals, so humans, organoids into animal chimeras, they fill with blood cells as normal. Now the way we do these organoids is that we have the world's first full human transcription factor library. This is over 1,700 ORFs, proteins that can be expressed in a variety of systems. Our favorite is piggyback, since it has the largest payload and the highest expression level. And we can do these in a multiplex manner. Remember, multiplex is one of the themes here, where we can check out the entire library at once in one droplet. And this is now spun off a company called GC Therapeutics that Alex and Peristu are spearheading. An example that we've put into mice for preclinical trials is a mixture of a number of different neuronal cell types, including oligodendrocytes and neuroprogenitors. These oligodendrocytes will wrap around the axons, just as they do in the white matter in the spinal column, to get rapid transmission, and it will do this with a normal number of wraps, the correct geometry which we quantitate, and it will, in a mouse demyelination disease model, it will restore function to normal levels from a score of 3 to a score of 4, relative to healthy mouse, a score of 4. So these are the oligodendrocyte precursor cell system. Another thing we can do with these human organoids is we can explore and develop therapies for late onset diseases. Now this was somewhat surprising to me, and I have to thank my collaborator Bruce Yankner for pushing us on this. We had already done accelerated development where we could get things that normally take hundreds of days in normal development. We can just cut to the chase and go in 4 days with 98% efficiency. But that's just making normal tissue. He felt we could also get pathological effects that normally take 70 years to develop. We could see some early effects, and we do indeed. In 2 to 4 weeks, we can see molecular and cellular anomalies that are quite significant, as you'll see in just a second, for late onset Alzheimer's, schizophrenia, and bipolar, and we're trying other ones now. So, for example, here's Alzheimer's, and this is published recently. You can take age-matched controls on the left for late onset Alzheimer's on the right, and you can see they're like night and day in this RNA transcriptome experiment. These are biclustering of the gene expression. But these differ, they're age-matched controls, but they differ by 3 million base pairs. You and I differ by 3 million base pairs, probably. So to do a really clean experiment, you need both the patient controls, case controls, and also changing one base pair. So we can do the epigenetic reprogramming to make these neuronal progenitors and brain organoids, and we can do epigenetic and genetic, by changing a single base pair with CRISPR or other editing tools, and can change, for example, ApoE4, which is about 20 times higher risk than the lowest risk ApoE. We can change it in one base pair, and again, we see this significant clustering. You don't need a roadmap to see how different that one base pair can make the expression. Now, putting transplanting into humans has suddenly changed. Since 1954, when we first started transplants, actually not too far from here, you've had to match donor. That is no longer the case. There are some successful cases. I use CAR T cells as an example, but it's spreading. This idea of universal donors requires editing to get rid of things that are, or to alter parts of the system that cause immune host versus graft or graft versus host. And these involve eliminating the T cell receptors if we're doing T cell population, as in CAR T, modifying the histocompatibility, for example, beta-2 microglobulin, and the PD-1 pathway. And here's one of the first recipients of these CAR T cells. I think this was zinc finger talon and CRISPR has been used clinically. Now take that one step further, from human to human to animal to human. And this is proposed over 20 years ago by some visionaries that later invited, once they saw that we had invented CRISPR, they invited us to help them, because initially it had been hoped that one gene, for example, the GGTA1, the alpha-1,3 galactosyl, would be sufficient, but then it became evident there were a lot of genes that had to be done. And we have a total of 43 genes that we've collected from everybody's wish list and then put them into a single pig. The first pig that we made was knocking out 25 endogenous retroviruses. This is because the FDA was correctly concerned that every cell of every pig in the world releases these viruses that are known to infect human cells and then reinfect and spread. And an immune-compromised patient, as is the case in almost all transplants, would be a breeding ground for zoonotic disease, like analogous to swine flu and Ebola and HIV. So that did not seem like a good thing. So we knocked that out, and it was actually very easy to knock it out and transform fibroblasts. It worked first time, which doesn't happen very often in our lab. But to get it to work in pigs, we had to use normal fibroblasts, and that took more effort, as you'll see in just a moment. But we have other parts of the system, clotting, complement, and the sugars, 43 different genes. But to get the multiple edits at once, if you don't do anything in normal cells are much more sensitive, and we've been working with normal cells since our very first CRISPR paper in 2013. If you don't do anything special, you get none that are completely edited, not even close. If you use basic fibroblast growth factor and p53 inhibitor, then you get almost everything is what you want. Now, so going from 25 to 43, well, from 2 edits to 25 to 43, our next logical step was 26,000. This is an exponential growth. And so we looked at the dark matter of the human genome, the part that nobody talks about in polite society because we don't have the genome finished, and it's hard to read these things, much less edit them. But if you want to address, so in the literature, there's good evidence for involvement in senescence, neurogenesis, cancer, inflammation. If you want to tackle that, and you want to knock out or alter every repeat, you need more powerful technology. So what we used was not CRISPR or any nuclease, because nucleases, even nicking, not double-strand breaks, just a nick, is extremely toxic at this level of editing where you're making lots of edits per genome. We had to use the most benign, non-nicking Deaminase, and our group and David Liu's group have pioneered this, where a single atom, a nitrogen, can be changed to an oxygen to change a C to a T or an A to a G. And here's examples in these line 1 elements in the human genome, of which there's about 100 active ones, but 26,000 that we ended up editing. And we had to do all these things that I've mentioned already to get it to work. This is work at Corey, Oscar, Khalid, and Farina. And here is what happened with normal human cells. So we now switched from pigs to humans, and these are personal genome project, actually from my body, since I am unlikely to withdraw from the project, as is You have to give an opt-out clause in the consent forms. We got 7,000 per these diploid cells with the A base editor, A to G transition. And then the C base editors are a little less efficient, and transformed cells are a little more efficient. So our record so far is the A base editor at 27,000 edits per cell. So that's a considerable step up from 2 edits per cell, which is where we were a few years ago. Now, the replacement of organs, the transplants, there's a transplant crisis, and that would be a sufficient goal. But one of the things that excites me about cell therapies and organ therapies is the possibility of enhancement. And enhancement is often taken off the table, I think, unjustly, but certainly for organs, you do not want your transplanted organs to succumb to pathogens or cancer senescence, immunity. You'd like to be able to cryopreserve them and protect them from DNA damage that might occur during, say, chemotherapy or space travel or so forth. None of these I think all of these are good ideas, and we know how to do them. There's at least some animal, natural or experimental, to illustrate each of these things, and I'll show you. I've shown some examples already having to do immunity, but I'll show you some more. Here's 2 animals, for example, that survive at liquid helium temperature and also desiccation. For the senescence, it's a kind of half-empty, half-full situation where we know a great deal about longevity and aging reversal in animals. These 9 major pathways have been supported time and again, and we are in the process of taking what's been learned and turning them into gene therapies. And we just had a paper come out in PNAS a few days ago. Noah Davison was the first author. And it started with this GeneAge database, which has been in use now. Pedro de Magalas developed it as a postdoc in my lab. It's been in use for over a decade. And we took the top 300 human genes and picked the subset, which are non-cell autonomous. So that's just jargon for, if you take something like telomerase, that just fixes the cell that you're in. But these genes will either be hormones or enzymes that are released from the cell into many adjacent cells. And that allows us to do experiments at the current state of less than ideal delivery. So we took the non-cell autonomous ones, we tested about 45 of them in mice, and then we went on to do, picked 3 of them that were the best individually and did them in every possible combination. And this combination gene therapy, I think, is a very powerful concept, and we so far on every disease of aging that we've tested, 5 so far, and this list will get longer, we see a significant impact, which implies or gives us hope that we can attack not specific symptoms of specific diseases, but the underlying core problem, which is that older cells and older people and animals don't repair, don't do a variety of homeostatic things as well as the younger ones. Most of us, if we don't intervene, will die of a disease that does not kill 20-year-olds, 90% of us. So these are some of the diseases that we've tackled so far, and this is now a startup company called Rejuvenate Bio that's aimed at dog therapies initially, because there is a market, and it is it's a better model for humans. Okay, and we are aiming at reversal rather than longevity, because longevity takes longer clinical trials, and in many ways that's not what we want. Anyway, we don't want to live a long time with sickness. We want to do preventative and reversal. To analyze both our organoids, which we would say are synthetic, they're normal analogs, and pathological tissues and synthetic pathological tissues, so I showed you we were simulating Alzheimer's, late onset Alzheimer's. We want to do this, we want to see how faithful they are, or in some cases where we're trying to make something that never existed before, how different it is, and we want to do this at the highest resolution with the most information-dense molecules. So these information molecules, DNA, RNA, and protein, we can now detect at up to 12 nanometer precision. So this is just microscopy, but it's 20/20 microscopy. So each pixel, each voxel in the image tells a story. So it gives a barcode. It tells you which DNA, RNA, or protein are there, and you can quantitate by counting, or the computer can. And this can be either confocal or super resolution. The cost of these things is actually less than the cost of a sequencer, and almost all, I should mention, fluorescent next-gen sequencers are essentially microscopes at their core. And the only difference between these next-gen sequencers or these microscopes and other microscopes and sequencers is that they combine the two, is they don't throw away the three-dimensional information, which is so valuable to pathologists. And so, for example, compared to a normal sequencer, our samples are 1,000 times thicker. Rather than a monolayer that's nanometer of randomized molecules, we take the molecules in their natural 3D configuration and thickness, and so for roughly the same reagent use and same instrument, we're getting 1,000 times more data and the 3D coordinates. So I'll show you in the next slide a single cell, single fibroblast, again from the Personal Genome Project. Each dot is a single RNA molecule, 4 colors for the 4 bases A, C, G, and T. They don't change position, just color, and then the computer can recite what it has learned. Remember, each pixel, each voxel tells a story. Each one is a barcode that tells you which RNA was at that position in the original single-cell fibroblasts. That's RNA. We can also do DNA and protein, and I'll show you that in just a second. Jay Lee is at ApplySci Harvard, and Evan Doherty helped start one of those 2 companies I mentioned, ReadCore, also Brooker. Now this is super resolution on DNA, and it doesn't make any sense to do DNA at less than super resolution because following the path of the chromosome requires that. This is work from Ting Wu's lab, professor at Harvard, and we have a shared collaborator, Peng Yin, and these are again the PGP chromosomes that help validate this using classical genetics. You can validate the super resolution here. Here's starting with the my mother and my father's chromosome 19 in my cells. You can walk through it, color coding each step that you take, and you can do multiple diploid chromosomes simultaneously. Their lab is moving towards doing all the chromosomes simultaneously for about almost exactly the same time and effort as doing just a little piece of one chromosome. So the computer can allow you to look at whatever you want. So this may finally allow us to actually sequence a whole human genome and do it very cost effectively and get the 3-dimensional coordinates all at once, because you're not breaking up the chromosome, you're not randomizing it, you're following it, tracing it from end to end. And finally, for the trifecta, I showed you RNA, DNA. Now this is protein. To do proteins, we use antibodies. The antibodies are labeled with nucleic acids to kind of convert it to a nucleic acid problem. This is a 9-mer, 9 long. And you can either use this big Y-shaped IgG, the classic antibody, or you can take one domain out of it, so-called nanobodies. And both of these can be, we can make large libraries and screen them for particular imaging tasks. These short DNA allows us to bind DNA, fluorescent labeled DNA, transiently, so you can get the centroids. The centroids are dozens of times higher resolution. This is called DNA exchange imaging. It's similar to other super resolution methods, but it's particularly appropriate for this DNA labeled antibodies. Here's an application to neuro, and this group is very much into neuro. Here we wanted to be able to trace the connectome, and the connectome is critically important to know where the synapses are, what direction they go from presynaptic to postsynaptic. So we have about a dozen antibodies that can help us tell not only what the direction of the synapses, but also whether they're excitatory or inhibitory. It is totally useless in a conventional microscope, which is diffraction limited, which everybody thought was the limit for many years. But now with this DNA exchange imaging, you can get into the 12 nanometer range. So just want to open it up for questions. These are some of the collaborators I mentioned. Jennifer Lewis helped with the with the vascularized organoids. Ting Wu has helped with has developed the super resolution for DNA. And then we've got a number of collaborators that work with us on synthetic biology for human called GP Wright. I was just at a GP Wright meeting in New York just yesterday. So thank you. Questions? Yes?
So I've been following tissue engineering for 30 years. See all the notable failures along the line. I personally feel like in the last 5 years that people are starting to make pretty significant progress. Could you, in your opinion, sort of share what you see if we were to flash forward 5, 10, 15 years? What maybe some of the winners and you know, contrast that a little bit with what has been done in the past and how things are moving in the future with respect to regen ed?
Yeah, so I think the reason it's different now is because these two exponential curves have finally kicked into tissues. I mean, these were kind of restricted to more basic, microbial synthetic biology and human diagnostics. And now our ability, this very steep exponential is kicking into developmental biology and so forth. And so that's one thing that's happening. The organoids are on their own exponential. We finally have the first complete transcription factor library that I mentioned. It's not yet publicly available. It's in Adgene, which is a nonprofit. It will be distributed soon. That they've totally done the QC on it. So that is another greatly enabling product. Our ability to image at super resolution allows us to really determine what constitutes a particular cell type. Once we know the cell atlas for the whole body, then we can use that, or even at our current partial knowledge, we can use that to make recipes for any cell type that might be missing from regenerative medicine. And then finally, the thing that's I think revolutionary is our ability to make these universal donors, in particular the pig donors, because that is very cost-effective regenerative medicine, because you can engineer the germline, which we can't engineer the germline of humans right at the moment, but you can for pigs, and you can, and they're a very inexpensive way of generating a very large number of organs that are potentially enhanced organs. So I think it's just a completely new ball game for all those reasons. Thanks for the question. It was good. Yeah.
The idea of using a person microscope to sequence and specimen, how long do you think it would take for these technologies to get samples at the level of a Deaminase sequencer? So pretty much preserving the 3D structure.
Right.
Together with the sequence.
Yeah, so the question is, how long will it take us to get a microscope that does both sequencing and 3D structure? I don't know for sure. I don't see any particular barrier. I mean, the uptake of these instruments has been quite rapid. We went from the ABI capillary sequencer to the 454 to the solid to the Celexa to the Illumina. Each of those transitions was less than a year. And so we're very fickle, I guess is what you could say, is we will jump onto the next technology and sequencing very quickly. Microscopy is a little more conservative, but not much. If you have a better microscope, and these are inexpensive desktop, completely automated, so you basically put your sample in, walk away, it'll process multiple samples for for days without intervention. So I think that, we've gotten much more comfortable with software packages that do a lot of the work for you. So I think it will be adopted quite quickly. It's already being adopted quite quickly. That's why we have examples of DNA, RNA, and protein. Each of those is a different group working on that. If you had to build it yourself, the adoption would be very slow. But there are now 2 instruments that are off the shelf from 2 different companies. So there's healthy competition. That's the other thing that sometimes slows things down, is you have a monopoly, which we do in sequencing, but we don't yet in situ sequencing. Yeah, sure. Yeah.
The concept of turning back the clock and sort of reversing or rejuvenating.
Yeah.
I always thought of aging as a systemic
Yes.
entire body, like you had said, some of the cells just don't behave like they do when you're a 15 or 20-year-old.
Yeah.
So could you talk to us a little bit about responding to individual organs or individual problems that or diseases that may occur that are localized versus a more systemic, whole system type aging process that's going on?
Yeah, so we face the issue of whether we want to go for specific organs or specific diseases, or we want to go for all of them. Aging impacts almost every disease, both common and rare. In fact, a lot of the so-called complex common diseases, I think, might be more fruitfully addressed by thinking about them as aging mostly, rather than thinking of them as some kind of GWAS problem where you have to have a complex genetics. I like the systemic approach. We're doubling down on it. We're developing new delivery mechanisms that will get us to every cell in the body. But in the meantime, we're using the non-cell autonomous route, where essentially it's like a drug, but rather than having to take the drug every day, inject the drug every day, your body is releasing. And these are natural drugs. It's basically during aging, a number of your very important proteins drop with age, and we're just boosting them right back up. So in a way, it's easier than most gene therapy, where you might be introducing a protein that is not in that particular individual due to mutation or whatever, and so you have an immune problem. Here we don't have an immune problem. So, so it's basically a once and done delivery, but we would like to get to systemic delivery where we can do non-cell autonomous things like telomerase and mitochondrial and things like that. Yeah.
Let's say you re-engineered your organ, one organ, and let's say you have discovered a set of genes that are going to either enhance or prevent the immune system from destroying the organ. But what about feedback mechanisms, in a systemic, in a system? Because let's say you have you've figured out all the genes, let's say, for a specific organ, but once you put it back into a human, for instance, there would be feedback problems on a systemic level.
So first of all, just be clear, we don't do one organ at a time. We're humanizing the entire pig so that we can take any of the organs. And in that sense, we're already making a system model that is, and I mentioned that we're doing complement, coagulation, sugars, all the sorts of things that would cause a system-level failure in the human. We're standing on the shoulders of giants. I mean, people have been working on all these individual systems, but still thinking of the whole system for 20 years. And all we did was we just satisfied their wish list by figuring out how to do multiple edits simultaneously with CRISPR. Doesn't mean that we will have that it will work first time. Oh, I forgot to mention, Jim Markman is heading a MGH preclinical trial, non-human primates, where we have 9-month survival so far. So it's a process of debugging. We want to use the same immune suppression drugs that are already approved for transplants. So we want it to be sometimes there's a temptation to use very powerful drugs that you can only use in animals. That to me, invalidates the clinical trial. But it's that sort of thinking about system level that you're trying to talk, that is what we're trying to do. And you can see where it fails, and because we have, essentially, we can manipulate any gene to be any pig gene or any human gene. It's amazing how quickly we can respond to these, the debugging feedback.
Okay.
Yeah, good question. Yeah.
Given the high throughput nature of all the data now that you're collecting on all these fronts, given the high throughput nature of all the techniques that you are developing to extract data from all these systems, how do you anticipate artificial intelligence actually being put into a table to actually improve all of this? I mean, it's obvious that it will, but how, I guess?
Yeah, I didn't mention it because of time constraints, but we have a large effort on machine learning, artificial intelligence, mostly focused on protein design, but also we're using similar tools for the image analysis. For the protein design, it was mind-boggling for me how well it worked, because I'm trained in 1970s technology, or ever since then, which has been energy and crystallographic 3D coordinates, and this machine learning can do it just based on sequence analysis and organ level things for which you don't have an energy model, and it just works spectacularly well. We've used it for engineering AAV, for example. We've made over 1 million synthetic AAVs, and not just machine intelligence, but this ability to do rapid prototyping with synthesis. You can think of that as a natural computing platform. It's kind of like a quantum computing platform, although not qubits, where you can make lots of prototypes. They give you the data back. So if you treat it as a black box, it's an incredibly powerful computing method. So that combination of machine learning and synthesis is a whole new way of doing computing.